Welcome to this explainer. Today we're cracking open the ultimate survival playbook. We're going to dive deep into the absolutely incredible biological hacks that single-celled organisms, plants, and simple animals use to survive, multiply, and thrive against all odds. Now, if you've ever felt totally overwhelmed by all those complicated biological terms, take a breath, don't worry. Today is all about conceptual clarity. We're tossing out the need for brute force memorization, and we're going to build your understanding from the ground up. Let's get into it.
All right, really quickly, here's our cognitive map for today. We're starting with the blueprint and the glitch, then moving through solo strategies, the regrowers, plant hacks, and finally wrapping up with spores.
Okay, let's dive into Lection 1, the blueprint and the glitch. Let's really look at why organisms reproduce in the first place.
Now, here's a kind of weird question to think about. Why do organisms even bother? Think about it. Things like nutrition, respiration, excretion. If an organism stops doing any of those, it dies, right? But if it stops reproducing, the individual is perfectly fine. Plus, making an entire copy of yourself takes a massive amount of energy. So why do it? Well, because survival is ultimately this massive high-stakes numbers game. If a species doesn't reproduce, it simply vanishes from the Earth. Reproduction isn't really about the individual at all. It's about the species maintaining its existence.
And at the very core of this entire survival game is a molecule I know you've heard of, DNA. I want you to imagine DNA as the ultimate master recipe book stored safely inside the kitchen, which in this case is the nucleus of your cell. These chromosomes contain the exact, precise instructions for baking the proteins that make up an entire organism. If you change the recipe, well, you bake a completely different protein, which ultimately leads to an entirely different body design. So, reproduction, at its absolute most basic level, is really just the act of copying this master recipe book.
So let's actually sketch this out mentally. Imagine you've successfully photocopied that master recipe book. Great, but you can't just toss that new copy out the window into the street and expect a cake to magically bake itself, right? That raw DNA copy has literally no machinery to process it. Because of that, copying DNA has to be accompanied by the cell building an entire second kitchen, a brand new cellular apparatus. Only when that entire new machinery is built can the DNA actually separate, effectively dividing one cell into two fully functioning cells. But are these copies absolutely 100% perfect? Well, no. No biochemical reaction is perfectly reliable. Sometimes there's a little glitch. Now, if the glitch or the variation is too drastic, the new DNA just won't work with its cellular machinery, and the cell simply dies. But what happens if that variation is just subtle? The new cell survives, but it's slightly different from its parent. We really shouldn't look at these glitches as mistakes. They're actually evolutionary superpowers. This inbuilt tendency for variation is exactly what allows a species to adapt and survive when their environment drastically changes over time. DNA copying is the absolute foundation of everything.
Moving right along to Section 2, Solo Strategies. Let's see how single-celled organisms actually pull this off.
Imagine we're stepping into the lab right now for Activity 7.3. I want you to mentally put your eye right up to that microscope lens. First, you look at a permanent slide of a regular, everyday amoeba. Then, you slide in a new one showing a process called binary fission. When you compare them side by side, you literally catch the organism right in the act of splitting its nucleus and its cytoplasm, creating two brand new individuals from just one. It's wild to see.
What's super interesting here is the contrast in how different things split. Look at our amoeba. It's essentially a shapeless blob, which means it can slice itself in half along absolutely any plane. It doesn't matter. But then we have Leishmania. Leishmania is the organism that actually causes Kala-Azar, or black fever, a really serious disease, but scientifically, a fascinating organism. Notice that little whip-like structure at the end of it? Because of that specific body organization, Leishmania doesn't have the freedom amoeba does. It's forced to divide in a very definite, highly structured orientation.
Now, both amoeba and Leishmania use binary fission, meaning they split into two. But what if you need to multiply, like, a lot faster? That brings us to multiple fission. The malarial parasite Plasmodium is a terrifyingly efficient example of this. Instead of dividing into two, it divides into many daughter cells simultaneously inside a protective wall, allowing it to basically explode its population all at once.
Let's scale things up just a bit to simple multicellular organisms. Picture Activity 7.4. If we go out and collect some of that dark green stringy algae from a pond, that's called spirogyra. We put those tiny green filaments on a slide. But crucially, before we put the cover slip on, we have to add a drop of glycerin. Why do we do that? Well, it prevents the specimen from drying out, keeping those delicate filaments crystal clear so we can actually see what's happening under the microscope.
And when you look through that lens, you see all the cells lined up perfectly. When Sparagyra matures, it doesn't do anything crazy complex. It literally just snaps into smaller pieces. This is a process called fragmentation. And here is where the magic happens. These broken fragments aren't just dead debris floating around. Because their multicellular body organization is so incredibly simple, meaning they don't have complex, specialized tissues, each little broken fragment just keeps right on dividing and grows into a brand new individual.
All right, Section 3, the regrowers. Let's look at regeneration and budding.
This right here brilliantly illustrates the absolute superpower of regeneration. Take the Planaria. If you chop this animal into 3 pieces, the pieces don't die. In fact, you could take some Planaria, cut them into 200 pieces, and you'd literally get 200 new worms. What happens is, specialized regenerative cells at the site of the cut start to proliferate rapidly. They multiply into this mass of cells, which then undergo a highly organized development to form completely new tissues and organs, eventually becoming a whole new Planaria.
Which naturally begs the question, if a chopped-up Planaria regrows effortlessly, why on earth can't a human regrow a chopped-off finger? Well, it all comes down to specialization. Complex animals are highly differentiated. Our skin cells only know how to make skin, our bone cells only make bone. We lack that localized pool of generic, highly proliferating cells that can suddenly just decide to rebuild a complex mix of nerves, blood vessels, and bone on command. Regeneration is amazing, for sure, but it is strictly limited by the complexity of the organism's design.
Another really cool way specialized cells do their thing is through budding. In animals like Hydra, regenerative cells multiply at one specific site, creating a literal bud on its side that grows into a tiny adult before finally popping off and detaching. And we see this in fungi too. Think back to Activity 7.1. If you take yeast, give it a food source, like a nice warm sugar solution, and observe it, you'll actually see those single yeast cells pushing out these little chain-like bubbles. Those are the buds separating and growing. It's a rapid, highly effective cloning process.
Moving on to Section 4, plant hacks. Let's talk about vegetative propagation.
Vegetative propagation is basically nature's ultimate cheat code for farming. Think about Activity 7.5 with the potato, or 7.6 with the money plant. If you cut a potato into pieces, only the pieces that contain a little notch or an eye will actually sprout green shoots, and that only happens if they're kept on perfectly moistened cotton. The water is what activates those dormant cells. So why do farmers absolutely love this trick? Because a plant grown from a root or a stem piece bears fruit so much faster than waiting around for a seed to sprout. Plus, every single new plant is an exact genetic clone of the parent.
Look closely at the edge of this leaf. Don't let the fancy scientific name Bryophyllum scare you. What it's doing is actually beautiful. Instead of relying on seeds, it naturally grows tiny, fully formed little plant buds right along the notches of its leaves. When those buds get heavy enough, they just drop off right onto the moist soil below and take root. The plant is literally just dropping ready-made clones of itself everywhere.
And we've actually taken this natural hack straight into the laboratory with something called tissue culture. Here, scientists scrape just a few cells from the growing tip of a plant and drop them into an artificial nutrient jelly. Those cells divide super rapidly into a clump that we call a callus. Then, by moving that callus into another jelly filled with growth hormones, we can trigger it to grow into hundreds of tiny little plantlets. It's a completely brilliant way to grow vast amounts of ornamental, 100% disease-free plants from just one single parent.
Finally, Section 5, spores. Let's look at some seriously resilient reproduction.
This brings us perfectly to Activity 7.2, observing rhizopus, better known as common bread mold. If you've ever seen an old piece of bread get all fuzzy and watched it spread like wildfire, this is exactly what's happening. Those little blobs on sticks are called sporangia, and packed inside them are hundreds of spores. The absolute genius of spores is their resilience. Each one is covered by a thick, tough wall that protects it from extreme heat, drying out, and really harsh environments. They just float around in the air until they land on something moist and nutritious, like your bread, and bam, then they germinate.
Wow, okay, look at this list. You literally just mastered the entire asexual playbook. You know exactly how amoeba split their fission, how Spiragyra snaps into fragments, how Planaria regenerates itself, how Hydra and yeast bud, how plants use vegetative propagation to hack the farming system, and how fungi use thick-walled spores to survive almost anything. And every single one of these strategies relies on one thing:
a single parent copying its DNA to create a brand new generation. But I want to leave you with one massive, kind of mind-bending question to ponder. We've just seen that asexual reproduction is incredibly fast. It is highly efficient. It requires 0 partners. So if this system works so perfectly for multiplying, why on earth did nature ever bother evolving complex, highly energy-consuming sexual reproduction in the first place? What advantage could possibly be gained by mixing DNA from 2 completely different individuals? Think back to that glitch in the DNA blueprint we talked about earlier. We're going to explore exactly how sexual reproduction supercharges that variation to drive evolution in our next explainer. Keep wondering, keep questioning, and I'll see you next time. Welcome to today's explainer. We're going to build complete conceptual mastery of literally one of the most fascinating processes in the entire universe. We are talking about how organisms reproduce and actually ensure the survival of their species. If you're studying biology right now, or honestly, if you're just curious about the brilliant mechanisms of life, you're exactly where you need to be. We're going to break down the atomic level logic and the real-world anatomy behind sexual reproduction. Okay, let's just dive right into it.
To get this all to make sense, we'll follow 6 logical steps today. First, why sexual reproduction? Second, reproduction in flowering plants. Third, human reproductive systems. Fourth, the unfertilized egg. Fifth, reproductive health. And finally, a quick chapter review and exercises.
All right, jumping right into part 1, why sexual reproduction?
Let's kick things off with a huge foundational puzzle about energy and survival. Think about it. Reproduction is actually not necessary to keep an individual alive, right? Not like eating or breathing. And if an organism can just make an exact copy of itself through asexual reproduction, which we totally see in organisms like Amoeba or Hydra, why do so many species bother spending immense amounts of energy just to find a mate? It seems kind of inefficient, doesn't it?
Well, here is the absolute brilliant secret of biology. It's all about variation. See, while asexual reproduction is super easy and fast, it produces exact DNA copies. So if the environment suddenly changes, say there's a massive temperature spike or some new disease out of nowhere, an entire population of identical clones could be wiped out instantly because none of them have the traits to survive. Sexual reproduction, on the other hand, combines DNA from 2 different individuals. This creates novel combinations, or variations. So even if conditions change drastically, these variations ensure that at least some individuals in the species will have the right genetic blueprint to survive and carry the torch forward.
But wait, combining DNA from 2 parents creates a massive mathematical problem. Imagine DNA as an instruction manual. If you take a full manual from the mother and a full manual from the father and just jam them together, the baby has double the pages. The next generation, 4 times the pages. It'd be a complete genetic traffic jam. To prevent this, nature uses a truly spectacular trick called meiosis. This is a special cell division in the reproductive organs that perfectly halves the number of chromosomes. So when the male and female germ cells finally combine, the new individual gets the exact normal amount of DNA. Problem solved?
Moving on to section 2, let's look at reproduction in flowering plants.
Now let's see this blueprint actually in action. If you've ever touched a hibiscus or a mustard flower and gotten a dusty yellowish powder on your hands, you've actually just touched the male reproductive cells. No way, right? That yellow powder is pollen, and it's produced by the male part of the flower called the stamen. Right in the center of the flower is the female part, the pistil. Its swollen bottom is the ovary, and that's what houses the female egg cells or ovules. The ultimate goal here? Get that yellow pollen grain down into the ovary. For fertilization to happen, that pollen has got to travel. This transfer is called pollination, and plants use 2 distinct strategies to pull this off. If the pollen simply drops from the stamen to the stigma of the exact same flower, that's self-pollination. Easy enough. But remember those survival-boosting genetic variations we just talked about? To get those, plants often prefer cross-pollination. This is transferring pollen to a completely different flower. But since plants can't just get up and walk over to each other, they need media, or agents, like a gust of wind, flowing water, or maybe a buzzing bee to act as their personal delivery service. So what happens after the pollen reaches the ovary and fertilizes the egg? It forms a zygote, which then develops a tough coat and becomes a seed. Let's do a quick mental whiteboard walkthrough of Activity 7.7 from the textbook. Imagine you take a Bengal gram, a chana seed, soak it in water overnight, and keep it wrapped in a wet cloth. The next day, you take a magnifying glass and carefully peel it open. What are you actually looking at? You are looking at a baby plant in suspended animation. You'll see a tiny, distinct top part called the plumule. That is the future shoot just waiting to grow up toward the sun. And then there's a tiny bottom part called the radicle, the future root waiting to dig down into the soil. It's literally the entire future of the plant packed into one tiny protective shell.
All right, let's step it up to Section 3, Human Reproductive Systems.
Let's apply this exact same biological logic to human beings and trace the map of the male reproductive system. The male germ cells, the sperms, are produced in the testes. Now, notice how the testes are located outside the main abdominal cavity, in a pouch called the scrotum. Why is that? Well, it's because sperm production requires a slightly lower temperature than normal body heat. Once formed, these sperms travel up a tube called the vas deferens. Along the way, it's kind of like they hit a biological drive-through. The prostate gland and seminal vesicles add special fluids to the mix. This fluid makes it incredibly easy for the sperm to swim, and it provides them with the vital nutrition they need for their really long journey out through the urethra and the penis.
Now, let's trace the female reproductive system. When a girl is born, her ovaries already contain thousands of immature eggs. At puberty, these start maturing. Every single month, one ovary releases a mature egg into a thin tube called the fallopian tube or oviduct. If sperms enter through the vaginal passage, they swim upwards to this fallopian tube. And this is absolutely crucial. The fallopian tube is exactly where fertilization happens. The fused egg, now called a zygote, then travels down and implants itself into the soft, elastic, bag-like structure known as the uterus. This is where it'll grow into a baby over 9 months, pulling nutrition directly from the mother's blood through a special tissue called the placenta.
Which brings us to Section 4, the unfertilized egg. But we've got to ask, what happens if that egg waiting in the fallopian tube doesn't actually meet a sperm? Well, think of the uterus as a really dedicated host, meticulously preparing a VIP room for a very important guest, a fertilized egg. Every single month, anticipating a baby, the uterus builds up this thick, spongy lining full of blood to nourish an embryo. But an unfertilized egg only lives for about 1 day. If no sperm arrives, the VIP room just isn't needed. So the body naturally resets. That thick, spongy lining slowly breaks down and comes out through the vagina as blood and mucus. This cycle takes roughly a month and lasts for about 2 to 8 days. And this completely natural, essential process is what we know as menstruation.
Okay, moving on to Section 5, Reproductive Health.
Because we humans understand the precise anatomy of this system, we can actually use science to control when reproduction happens. To prevent unwanted pregnancies, we use contraceptive methods. And these fall into 3 beautifully logical categories based on the exact anatomy we just learned. First up, mechanical methods. This is where you simply create a physical roadblock. Things like condoms or a copper T placed in the uterus physically prevent the sperm from ever reaching the egg. Second, hormonal methods. Oral pills actually change the hormonal balance in the female body, meaning an egg is never even released from the ovary. No egg, no fertilization. And third, surgical methods. Doctors can do a minor procedure to block the vas deferens in males or block the fallopian tubes in females. This permanently cuts off the transport routes altogether.
Finally, Section 6, Chapter Review and Exercises.
You've just mastered the core concepts, so let's lock them in really quick. Think back to the map we just traced together. The egg is made in the ovary, and the baby grows in the elastic uterus. But where do the sperm and egg actually meet? If you answered the fallopian tube, you are absolutely correct. That right there is the specific site where the magical fusion of DNA finally happens.
And this brilliantly illustrates our main takeaway for today's explainer. Whether it's pollen riding the wind or human DNA literally having itself to combine perfectly in a fallopian tube, sexual reproduction is this incredibly complex blueprint entirely designed to create variation. And that variation is the ultimate shield that ensures survival. But it leaves us with a pretty fascinating question to think about. As our planet's environment begins to change faster than ever before, will this built-in biological shield of variation be enough to help species evolve and survive the future? Keep asking questions, keep exploring the amazing science of life, and I'll catch you in the next one.
All right, let's jump right into this board exam masterclass for Biology Chapter 7. Listen, if you're aiming for top marks in the Karnataka SSLC exams, knowing the science behind how do organisms reproduce just isn't enough. You need to know exactly how your paper gets graded. So, in this explainer, I'm not talking to you as a teacher. I'm stepping into the shoes of a veteran board exam evaluator to show you exactly where you win and lose those crucial marks.
I really want you to adopt this veteran evaluator mindset right out of the gate.
Look, when we're sitting there with thousands of papers to grade, we honestly don't care about your word count. We're not looking for a novel. We are literally just scanning your answers for specific keywords and value points. If you hit the keyword, boom, half mark. But if you write this beautiful, scientifically accurate paragraph and miss the exact keyword from our official rubric, you lose the mark. It's really that simple, and yeah, it's that strict. So, here's our game plan for today. We're going to cover the evaluator's blueprint, 1-mark multiple choice traps, diagram masterclasses, the human anatomy score breakdown, and finally, those placenta and contraception gold mines.
Okay, part 1, the evaluator's blueprint. Let's peel back the curtain and see how that grading rubric actually works behind the scenes. This right here perfectly illustrates the golden rule of diagrams. Whenever you look at the official grading scheme, whether it's the human heart or a reproductive organ, the point distribution is identical. You get 3 solid marks for the sketch itself and strictly half a mark for each correct label. So think about it. If a question only asks for 2 labels, maybe the septum and the right atrium, that is exactly 1 mark total for labeling. Please do not waste precious exam time drawing 10 different labels if they only ask for 2. Give the examiner exactly what they ask for and secure that 4 out of 4.
Moving on to part 2, 1-mark multiple choice traps. You know, beyond the drawings, the board absolutely loves setting little traps in the multiple choice section. This one is an absolute favorite for the examiners. Every single year, students mess up the exact sequence of sexual reproduction in a flower. You'll get a multiple choice question asking for the right order, and the trap is that they'll place seed before embryo, or fertilization before pollination. So here's your insider secret. Just use the mnemonic PFES. Pollination happens first, followed by fertilization, which creates the embryo, which ultimately becomes the seed. PFES. Lock that in your brain right now.
Next up is this classic genetics trap. The official paper will ask something like, in a zygote, there are 23 pairs of chromosomes. So, the number of chromosomes present in the normal cell of the parent organism is, and students instantly panic. They think, oh wait, a zygote comes from 2 parents, so I got to double it to 46 pairs, or they try to divide it in half. No. Here's the crucial point. A normal human parent cell has exactly 23 pairs, the gametes have 23 single chromosomes, and they combine to restore the 23 pairs in the zygote. So the answer is just 23 pairs. Don't let their sneaky phrasing trick you into doing math you just don't need to do.
And then there's this one. If you see a seedling diagram where an arrow points to a part growing downwards, please don't just write, it grows towards the earth. I mean, that's a total novice answer. The official key strictly requires the exact scientific keyword. You absolutely must write positive geotropism. Don't throw away an easy mark here. Just give the evaluator the exact terminology they're scanning for.
All right, let's jump into part 3, diagram masterclasses. Let's see how this strict rubric mindset builds your score in the 2-mark and 3-mark sections.
Take the germination of pollen on a stigma, for example. This is a massive high-yield target. The key answer here specifically awards 1.5 marks for getting the main structure right and exactly half a mark just for drawing an arrow pointing to the pollen tube. Now, the common student mistake here is drawing the pollen tube so it just kind of stops halfway down the style. Evaluators are specifically checking to ensure your pollen tube actually travels all the way down to the ovary to meet the ovule. Draw it completely, label it clearly, and bag those 2 marks.
Now, what about when they ask what happens in a flower after fertilization? Do not write a long, winding story. Seriously, don't do it. The evaluator has a rubric with exactly 4 half-mark bullet points. You just need to give them these exact 4. Number 1, the zygote divides into an embryo. Number 2, the ovule develops a tough coat to form a seed. Number 3, the ovary grows and ripens to form a fruit. And number 4, the petals, sepals, and stamens fall off. 4 quick bullets, 2 full marks. Boom. Done.
Okay, part 4, human anatomy score breakdown. This is a section where students routinely mix up functions, and it costs them those vital half marks that separate the A grades from the A+ grades.
You really need to explicitly avoid this trap when asked about the female reproductive system. They'll ask for the distinct functions of the ovary and the fallopian tube. Now, the ovary produces the egg or secretes hormones. That gets you your first mark. But the fallopian tube is the actual site of fertilization. You could also write that it carries the egg from the ovary to the womb. If you accidentally say fertilization happens in the ovary or the uterus, you're getting a strict 0 for that part. You've got to be exact.
And for the male reproductive system, structure your 2-mark answers strictly to separate the functions. The vas deferens is simply about transport. It carries sperms to the urethra. That's 1 mark.
But for the prostate gland and seminal vesicles, there's a dual requirement in the rubric. Their secretion helps transport the sperms, and it provides nutrition. If you only write "it makes semen" or "it helps transport," you might only get half a mark. You have to mention both transport and nutrition to lock that mark in.
Which brings us to our final section. Part 5, Placenta and Contraception Gold Mines. This right here is where we secure your top-tier grades by cracking the 4 and 5 mark-long answers.
Listen, the placenta question is almost guaranteed to show up. It usually asks you to explain the structure and important role of the placenta. For structure, you mention it's a special disc-like tissue embedded in the uterine wall. But for those function marks, you must show the two-way street. You have to mention the inward flow, providing a large surface area for oxygen and glucose to pass from the mother to the embryo. And you have to mention the outward flow, removing waste substances from the developing embryo into the mother's blood. Inward nutrients, outward waste. That is your strict 2-mark rubric right there.
Another absolute goldmine is contraception. If the paper asks for 3 methods of contraception for 3 marks, don't just list 3 random things. Structure your answer. List the scientific category, followed by one specific example. So you'd say barrier method, like condoms. Intrauterine devices, or IUDs, like placing a copper T in the uterus. Chemical methods, like oral pills. Or surgical methods, like blocking the vas deferens or fallopian tube. Categorize and conquer. It makes it incredibly easy for the evaluator to just tick, tick, tick and award you full marks.
So I'll leave you with this final question. Are you just studying the science, or are you finally ready to write exactly what the board rubric demands? You now know exactly where those hidden half marks live, and you know the traps the examiners are going to set. You have the blueprint. The only question left is, will you use it to write the perfect exam paper?